Hello everyone. I'm going to go through the first page of the single strand DNA mutations worksheet to help get you started. We're going to first go through the instructions. I'm going to go over the original gene sequence with you, and then we're going to do the first deletion together. In this worksheet, we are going to be going from DNA to messenger RNA to amino acids. So we are not including anticodons. We've done this for a little bit. We've been doing a lot of practice. And so in this particular worksheet, we're learning how mutations in the DNA affect the amino acid sequence. And when the amino acid sequence is affected, the functionality of the protein is also affected. Some proteins work incorrectly. Some proteins come out so small that the body just breaks them down. And so there's different variations. So we're going to be looking at the different types of mutations by looking at single DNA strands. So let's go ahead and read the instructions. It says, you have been given a single strand of DNA which has 10 codons. Remember, every 3 letters is a codon. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 codons. This strand represents the perfect gene with no mistakes. You will use this strand to compare what happens during different types of mutations. So before we can start looking at the mutations, we first need to know what the amino acids look like in a perfect strand. So the first thing we're going to do is we are going to transcribe the DNA code into messenger RNA. So the T becomes A becomes U, C becomes G, G becomes C, C becomes G, G becomes C, T becomes A, T becomes A becomes U, A becomes U, C becomes G, A becomes U, C becomes G, G becomes C, C becomes G, C becomes G, G becomes C, C, G, G, C, C, G, A, U, G, C, C, G, A, U, T, A, U, T, A, C, G. All right, so we have all 10 codons. And remember, when you're writing this, you always want to keep the codons, okay? Because every codon will code for an amino acid. So now we're moving on to the amino acids. AUG. So here I have a codon chart, okay? First letter is A, second letter is U, so that takes us to this box. Last letter is G, so met, which is methionine. So I'm going to go ahead and write met on my paper. This is the first amino acid. My second amino acid is codon is CGC. C, G, C, ARG, arginine. The third is AAU, AAU, ASN. The fourth is UGU, UGU, CYS. GCG, GCG, ALA. And we have another one, GCG, ALA. Okay. CGU, CGU, ARG. And again, CGU, ARG. UAU, UAU, TYR. And then UAG, UAG, stop. Okay, so this is our amino acid. This represents the amino acid chain, otherwise known as a polypeptide chain, which will eventually become a protein with no mistakes. This is the perfect gene. So I'm going to write that over here. Perfect gene. This is the gene we are going to use to compare all of our other problems to. We're going to compare number 1 to this. We are going to compare number 2 to this. We're going to compare number 3 to this. You are also going to compare number 4 and 5 to this sequence. Okay? So when it asks you how your mutated gene compares to the original, you're always going to come back and look at the original in order to formulate your answer. All right, so we're going to do the first one together. We've already gotten our perfect gene, so now we're going to do number 1. The first mutation you will need to model is deletion mutation. Look at the original strand and delete the 7th, 8th, 9th, and 10th nucleotides. Now, it says nucleotides, not codons. Nucleotides are the individual letters. Codons are the 3-letter words. So we need to look for 7, 8, 9, and 10th nucleotide. So we're counting 1, 2, 3, 4, 5, 6, 7 we have to cross out. 8 is going to be crossed out. 9 is going to be crossed out. And 10 is going to be crossed out. So it says the original gene sequence, we crossed out 7, 8, 9, and 10. Now it says rewrite the mutated sequence. So now we're going to rewrite this with these missing, with these deleted. So TAC, GCG, and we're going to skip all the way over here. The next 3 is going to be CAC. Okay, and you can cross it out to help you. GCC, because we still have to do it in codons and triplets. GCG, CAG, CAA, TAA, and now we're left with TC at the end. And there's nothing we're going to be able to do with that TC at the end. Okay? It's not going to code for anything. Now, before we start to do the transcribing and translating, let's look at how deleting those 4 nucleotides changed the number of codons. Originally, we had 10 codons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I pointed at the amino acids, but there's 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Because we deleted 4 nucleotides, we now have 1, 2, 3, 4, 5, 6, 7, 8 complete codons. So we have lost 2 full codons. All right, we're going to go ahead now and transcribe into mRNA. T becomes A, AU, CG, GC, CG, GC, CG, AU, CG, GC, CG, CG, GC, CG, GC, CG, AU, GC, CG, AU, AU, TA, AU, AU, AG. Okay. Now we're going to go ahead and translate into an amino acid so that we can see how these amino acids are different from the original perfect gene. So AUG, and by now we should know this one by heart. AUG is met. CGC. CGC ARG. GUG. GUG VAL. CGG. CGG ARG. CGC. CGC ARG. GUC. GUC VAL. GUU. GUU VAL. AUU. AUU ILE. AG. Nothing. We're just going to draw a line. Okay. There's nothing we can do with that. Now the question says, how did the mutated gene change from the original strand of DNA? How was the polypeptide chain affected? So how did it change? Well, we talked a little bit already. We went from having 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 codons to only having 1, 2, 3, so 1, 2, 3, 4, 5, 6, 7, 8 complete codons. So that's the first thing we're going to write. The original DNA strand has 10 codons. After the deletion, the mutated DNA only has 8 complete codons. Okay, that's the first thing. Now let's look at the amino acids. So the first amino acid in both polypeptide chains is MET. The second is ARG. But the third, ASN to VAL. And the fourth, CYS, the new one is ARG. The fifth, ALA, it's now ARG. The sixth, ALA, is now VAL. The seventh, ARG, VAL. The eighth, ARG, ILE. And then, of course, we don't have those two. So we can say that the third codon on are all different in the mutated strand because at the third codon, there is a change. And all the amino acids following that are different. So the third codon in the mutated DNA is different from the original DNA. Codons 3 through 8. I guess you could write either the third codon. I think I'm going to erase that. Hopefully it erases nicely. Okay. Let me rewrite that. We're just going to write out the codons that are different. I think that'll make more sense. Codons 3 through 8 are different in the mutated DNA strand from the original DNA strand. Okay? And that's it. All right? And now here, I'll put it up so you guys can see it better. So that's our complete answer. Okay? The original DNA strand has 10 codons. After the deletion, the mutated DNA only has 8 complete codons. Codons 3 through 8 are different in the mutated DNA strand from the original DNA strand. Okay? And if you want to, you can also number them if that helps you. Okay? You can number them. That way you know exactly what number to talk about. You just want to make sure that you're numbering correctly. Okay? Another important difference is that the mutated strand does not have a stop codon, but the original does. So that's another thing you can add. I'll go ahead and write it down here. The mutated strand does not have a stop codon at the end. Okay, that's another thing. So as you can see, by deleting 4 nucleotides, we have at least 3 changes. And that's not even considering how the polypeptide chain will fold differently and function differently. All right, so you guys are going to go ahead and continue on to doing number 2, number 3, number 4, number 5. Make sure you always compare them to the original. And of course, answer this back one, and there's a bonus. I will see you tomorrow.